Okay, class, so we're back to looking at phototransduction and capturing light. We spoke of before the pigment synthesis, rhodopsin forms, accumulates. So rhodopsin forms and accumulates in the dark. When the light occurs, there's bleaching and it separates. Pigment bleaching is when rhodopsin absorbs light, retinal changes to the all-trans form, and retinal is separate. And then the pigment regeneration we spoke of using ATP. Okay. I would like for you to look at this particular video. I thought it was very good. A video that I saw that I liked, and that gives you a nice description of the activity of this phototransduction. But anyway, let's continue on. Okay. So what actually happens then is this. I'm going to go here. Let's look at the activities that's going to occur in this particular aspect. All right. So light comes in. Now the key to this, let me just say this. The key to this are these channels here. The key to this are these channels. These are cation channels that allow sodium and calcium in to the membrane here of the rod. When they allow this in, then that causes a certain polarization on the membrane at a negative 40 millivolts. Now you may want to go back. I did a pretty extensive thing on electrophysiology, and you may want to go back. But we have a negative 40 millivolts in this membrane here with these coming in. Okay, if we were to close these off, then positives would not be coming in. But the potassium, as I alluded to, coming out, combined with the large molecular anions, would keep the membrane potential, let's say, would drop the membrane potential because what would happen, you wouldn't have positives coming in, but you would have them going out, combined with the large molecular anions. Go back again and look at electrophysiology. So the key to this is this. All right, the second key to this is this. What's keeping this open is a cyclic GMP molecule. What's keeping this open is cyclic GMP. So if you break down the cyclic GMP, this door closes. So again, let me go back again because it's quite detailed. The key to this whole thing is to keep this open, and this occurs during darkness. To activate it, though, you have to close the positive ions coming in, and it has to go hyperpolarization. So right here is polarized at a negative 40. If I close this off, then I'm not bringing positives in, but due to the potassium exiting and the large molecular anions, but I'm not bringing positive in, it's going to go more negative to around a negative 70 hyperpolarization. So it's the hyperpolarization that's going to cause the activation and the perception of light within the rods. So that's the key. The key is to remove cyclic GMP, close this, don't let the positive in, and go hyperpolarization. Bottom line. I want you to read on it, but you also can look at the video. Okay. So let's go back. Let's go back now that we've said what we have. Okay. So here is your rhodopsin. Let's assume that this is in the cis form, the form that allows the retinal to attach to the opsin. Okay? Light hits this. When light hits this, it changes from the 11 cis form to the all trans. When it attaches to the all trans, this separates. The separation of that allows then this to to attach and activate with something called G protein. Now we're into secondary messengers. We're into secondary messengers now. Secondary messengers carry signals through a cascade reaction. You attach to something at a membrane, the membrane activates, so a secondary messenger is like a butler. You go to a very wealthy person's house. The person doesn't come out, the butler does, and you tell him I'd like to see him. Then the butler carries the signal in to the person. So that's what a secondary messenger. That's what a secondary messenger is. So the secondary messenger carries the signal in, but the secondary messenger aspect also is an amplification process. So the first messenger is the light. The light then turns on the second messenger to carry the signal into the interior of this particular disc here. Okay, that's the secondary messenger. But a secondary messenger is an amplification process where the initial activity through these series of events amplifies the activity. So let's go back again. The light comes in this membrane here of this disc is the photopigment. The photopigment picks up the light. Okay, the retinal does, it picks up the light. When it picks up the light, that changes the configuration of the 11 cis form to the all trans form. Now, inside the membrane here is something called G protein that's there, a G protein. G protein cannot attach to the rhodopsin as long as the retinal is attached. But once the retinal is removed, and what removes it is when it becomes all trans retinal, it can no longer attach to the opsin. Okay, so now it can attach. This then activates the G protein. The G protein then can go and combine with something called phosphodiesterase. Phosphodiesterase is an enzyme that can break down cyclic GMP. It can break down cyclic GMP into GMP. Once you break down the cyclic GMP, remember it was the cyclic GMP that kept the channel open for the sodium and calcium. When you break it down, then you close this, and this goes then to hyperpolarization. The hyperpolarization at a -70 takes the story further as we look at it here. Okay, here is the outer segment of the rod. Here is sodium and calcium being able to enter. This is the dark reaction. The cyclic GMP gated channels are open because the cyclic GMP keeps them open. This is in the dark now. The voted gated calcium channels open synaptic terminals. Okay, so what happens is when the cyclic GMP is there, sodium and potassium can enter, sodium and calcium can enter, keeping this at a -40 millivolts, -40. What that allows then is the release here onto the bipolar cell that allows opening of calcium gates. The calcium comes in, and the calcium that comes in causes the release of glutamate. Glutamate is an inhibitory synaptic neurotransmitter, IPSP. Go back to my electrophysiology. That means that turns off the bipolar cell, disallowing it from releasing its neurotransmitter onto the ganglion cell. Now, all of this is what we call a graded reaction, which means it's not an action potential. Go back again to my electrophysiology. Action potential is all or none. So depending on various things of stimulation, when we get to the light aspect, that determines how much intensity that you will get as far as to BC bright light versus dim light, shall I say. So the neurotransmitter glutamate gives an IPSP at the bipolar cell, hyperpolarizes that. Remember, an IPSP is due to either a gate opening to chloride, or which would come in and make it more negative, or opening to potassium to let it out to make it more negative, pulling it further away from a threshold. The ganglion cell can do action potentials. All of these, everything here up to this point is graded. This is the one that does the action potential and carries it into the brain. So the hyper closes voltage gated calcium channels, inhibiting neurotransmitter right here because of the IPSP. So you can't turn this on just to make this try to reach threshold. No EPSP. Okay, so we go to the next one now. In the light reaction, what will have happened, going back to what we already talked about, the light would then come in. It would cause the change from the 11 cis form of retinal to the 11 trans form. The 11 trans form then would dissociate from the opsin. Once it dissociates from the opsin, then the opsin can attach to the G protein, transducin. The transducin then, once that happens, can cause the activation of phosphodiesterase. Phosphodiesterase then would break down the cyclic GMP, which kept the gates open to sodium and calcium, and cause them to close. When that closes, then you won't have as much positive stuff coming in, but because of potassium going out and large molecular anions, which keep a resting membrane potential, this would then go to a hyperpolarizer, a -70. A -70 here causes no neurotransmitter to be released. So the glutamate is not released here. When the glutamate is not released, then that allows this to now do an EPSP, excitatory postsynaptic. When that occurs, then calcium can come in here and cause release of neurotransmitter on the ganglion cell. And once that occurs, then action potentials can carry this into the brain. So this would be the light reaction that you would happen. So when the light hyperpolarized stop releasing inhibitory, which is glutamate, bipolar cells no longer inhibited, and action potentials. But up to here, everything is graded. Here is all or none in the ganglion cell. Okay. So that's what happens there. And going back again, though I am talking on this particular aspect here, go here, the same thing, even though we're doing this one, the same thing actually happens in the cones too. Now, one thing too, so here's some terms: rhodopsin, cation channels, transducin. Arrestin you didn't see. What arrestin does is, and I'm going to go back here, what arrestin does is that once this here is phosphorylated, once you phosphorylate the opsin, then arrestin can come in on the phosphorylated opsin and keep it from not any longer activating for a moment. Okay? And glutamate is the inhibitory. So we go back. We've spoken of that. This. Bipolar cells. We talked that. I go here. And I'm going to go on down to here, which we did the light reaction. Just going through everything. And that's where we are there. I'll go a little further. And we have now covered everything we need to cover on this particular aspect. Okay? So light adaptation we'll get to in a moment. Okay?